Pritzker lectureship this morning. The Pritzker Distinguished Lecturer is Mike Shuler. Mike is the James and Marsha McCormick Chair of the Department of Biomedical Engineering, as well as the Samuel Eckert Professor of Chemical Engineering in the School of Chemical and Biomolecular Engineering at Cornell University. Mike got his undergraduate degree from Notre Dame in 1969 in chemical engineering and his PhD from the University of Minnesota in 1973. He joined the Faculty of Chemical Engineering at Cornell in 1974 and rose to the position of chair from 1998 to 2002. In 2004, he became the founding chair of the Department of Biomedical Engineering there, and he's advised more than 50 PhD students and numerous master's students and 17 different postdocs. Mike's research is focused on biomolecular engineering and nano-biotechnology with an emphasis on applications in human health. His work involves the development of what he calls, in quotes, a body on a chip, which is a micro-fabricated, micro-fluidic system including cells and tissue constructs representing and emulating the various tissue organs of the body. I mean, I would say in passing that this research has the potential to revolutionize the ways in which we develop new drugs and new procedures at significantly lower cost, and has a tremendous potential for impacting in a good way the cost of healthcare in our country. So without further ado, I'd like to call Mike up to the stage and invite him to speak.
It's certainly a great honor and to be chosen to give the Pritzker lecture. And I want to thank many people involved. Mike King, I know, sent in the nomination. I didn't know he sent it in until after he sent it in. And I actually don't know all the people I need to thank that wrote letters. But I really very much appreciate it. This is really a great honor for me. Also, I want to thank the students that worked with me over the years. Between the master's students, the thesis, PhD students, and postdocs, it's over 100 students, and then I have numerous other students that did master engineering or undergraduate work with us. So I can't thank all of them individually, but it's been a true collaboration. The ideas that we have brought forward have been collaborative between myself and the students. So many of the ideas that we're talking about are really ones that were generated in collaboration with my students. I want to talk a little bit about modeling life, something I've thought about ever since I was a graduate student. And there's two kinds of models I'm going to talk about. One, I'm going to talk a little bit about mathematical or computer models. I want to test the completeness of understanding what is essential to be alive. What does it mean to be alive? Can we capture that in a model? Way to test what we really know. Then also, and again, you heard Rick mention a little bit about a body and the chip work. We're looking at building a surrogate for in vivo systems that we think can aid in the drug discovery, drug development process. Also offer another opportunity for us to do estimates of environmental toxicology. So I'm going to talk about three models. I'm going to talk about one which we started way back when the idea came when I was a graduate student, was simple bacterium, E. coli, followed by a little work we've done on what we call a minimal cell, kind of a portion of synthetic biology, artificial life, and then this body on a chip concept, which instead of a mathematical model is more a physical model. So the idea for building these realistic cell models really something that happened when I was a graduate student. I worked with Gus Ayers and Henry Tashia as my advisors, both of whom have passed away, and Arnie Fredrickson also was very influential at that point. What we thought about was different than people had been doing at that time, was thinking about building models of cells where we actually use the real concentration, intracellular concentrations. And I focused on a single cell. All these were rather unique at the time. This is an example of one of the single cell models that we built. It's a lumped parameter model, but it requires that you think of different components all moving on the same time scale and the same ratio. It actually turns out to be a fairly effective way to model a cell. We got up to about 34 different components, and we could mimic many of the different things that happen in a living cell. One of the things that makes us different from other previous cell models was there's no artificially imposed constraints. We didn't say the cell had to have a certain growth rate, had to be a certain size, shape. All these were outputs of the model rather than input of the model. Simply once we established the parameters and the structure of the model told us what all these things had to be. The model could respond to a wide range of responses. It could, for example, predict transient behavior fairly accurately. We used it, for example, to look at the response to genetic manipulation, particularly early on in terms of adding plasmids, looking at production plasmid-coated proteins, which you could do in a fairly robust way. It includes cell geometry, control of chromosome replication, mimic cell division cycle. Again, the cell geometry is an output of the model, not an input. So this is a coarse-grained approach where we lump everything together. And one other thing that makes this a little bit different than models that a lot of people are creating is that there are a lot of events that occur. A lot of events occur in a cell cycle that you have to take into account. And this has an effect on how you put the model together. An example of an event would be initiation of DNA replication or cell division. And this could be done in a modular approach. We could take a component of the cell and blow it up in genomic and chemical detail, leaving it in the structure, which was the structure was coarse-grained. And we could then relate changes at the genomic level to the overall physiology of the system. And we did that for nucleotides, and we did it for lipids and DNA replication. So what this has done is allowed us over many, many years, we worked on this for 3 or 4 decades, is to apply. We could predict general growth trends fairly accurately. We could predict what happened if you did certain manipulations to the cell. We could predict what happened if you made certain changes in the environment. The fact is modular allowed us to hone in on detail in certain components of the system while leaving the rest of the system in a more tractable form. And again, because it's modular, it could incorporate specifically genomic information. Now, if you know E. coli, there's lots of things in E. coli we don't know about. A lot of genes that are there that can code proteins, we just don't know what they do. So we can't actually build a rationally complete total model of E. coli, and that's why this coarse-grained mold works. But about 10 years ago, there was a lot of emphasis on artificial life, synthetic biology. One of the things that appealed to us was the design and construction of an artificial cell, and one that was really human design, as a platform, for example, for producing proteins or detoxifying waste or something like that. And actually, in terms of at least the protein production, something we thought of a long time ago. One thing I talked about when I had the opportunity to give the Johnson lecture at the American Chemical Society. So artificial cell, there's 2 approaches. The bottom one, biotechnological platform cell. It's more or less what we thought about back when I was doing the Johnson lecture. And the minimal cell is actually asking the question a somewhat different way, and that is, if I were designing a cell from scratch, what would be the fewest number of genes that I would need to make that cell function? How are these genes organized? How is their expression regulated? And of course, the minimal cell is going to be a dumb cell, right? Because it's the fewest number of genes, so it's going to have limited robustness. So we're also interested in the degree of robustness it might have. So why would we do this? One is simply, we want to understand better the basic design rules of life. If you were in the position to design a cell, what do you really have to worry about? What's the minimum regulatory structure that you need? If I have genes, how should they be organized? There were people much braver than I, like Craig Venter, that were talking about constructing the minimal cell themselves experimentally, and such a model could provide a method to guide experiments to construct such a cell. One of the things we found that I think was not appreciated by the Venter group is that the initial conditions inside the cell will make an influence on whether it's successful or not. So even if you got the genome correct, you have to be better than a cell in which the initial conditions are permissive for that cell to grow and sustain itself. And I think they had some problems that way that they didn't realize. Also, if we had such a model that could serve as a basis for this biotechnological platform cell I talked about, and evaluation of alternative design strategies for such a cell. It's more than a metabolic model. There are a lot of metabolic models, but it has to contain much more. It has to contain all the functionality in the cell. Things like those events I talked about, and control of cell replication, what size is the cell, the physical and chemical constraints within inside the cell. So reconstructing life in silico in the computer, where we're asking what are necessary building blocks, and how to connect physiology with genomics. As a side note, there's a lot of interest in pathway engineering, metabolic engineering. You take a pathway, you engineer it, see what comes on out. And this is pointed out by Jay Bailey almost 20 years ago. That approach gives you a correct answer only if you assume that the output of the pathway can never influence the input back into the pathway. And once you put a pathway inside a whole cell, there are lots of things that can happen, either in terms of secondary or tertiary effects, which influence input back into the pathway. So doing metabolic analysis without doing it in the context of the whole cell can lead you to incorrect conclusions. So our strategy for a minimal cell. One, we're going to use the E. coli model I talked about before as a framework in which to construct this minimal cell. The E. coli model was physiologically complete. In other words, I had 36 components, but I lumped everything together, so I didn't leave a component out to make it. And it has events, so it's much more than just a metabolic model. And we used a minimal gene set suggested by Gill et al. We modified it slightly because we had a couple of disagreements about which things to be included or not include, but that's the basic framework we went from. So we ended up a model which had 241 genes, 408 chemical species, a lot of reaction parameters, and 36 events. Now that looks like a lot of parameters to estimate. It's not as bad as it looks. Okay. First of all, it's a hypothetical cell, so all we need to do is have a set of parameters that work. But there's a strong constraint here, which if you know the composition, initial composition of the cell, we can guess what would be a reasonable initial composition for a cell. We know that at the time it divides, I have to have twice as much as what I began with, the steady state. That puts a very strong constraint on the parameters. That and the fact that no concentration can go less than 0. Those two constraints actually make it much easier to come up with estimates of reaction parameters than one might guess. The other thing to realize is that what's important is the ratio of one parameter to another. So the absolute values determine the growth rate, but it's actually the ratio that determines essentially the concentrations. So if realizing those constraints, it's actually much easier to come up with a set of reaction parameters than one might guess. You can almost do a lot of them by hand. So can we build a minimal cell model that can achieve stable replication? Can we have a model where the cell will divide for 1,000 generations? That was our goal. We assumed that it did 1,000 generations, it'd do 1 million. Okay. We're interested in things like response to gene knockdowns and gene knockouts, because we're interested about which genes are essential and the robustness of the resulting model. So this is an example. We made our initial guesses of cell size, geometry, composition. We begin the model. As long as we're close enough, it will tell us what the steady state is. Okay, if we're far enough off, it won't, but if we're close enough, it'll get us there. And this is just an example in terms of coming up to a steady state in terms of the cell geometry. And after, this is after 15 hours, it came to a relatively stable situation in terms of replicating levels of different enzymes. There actually is some subtle long-term variation which takes place longer than a single generation. And if you look closely, you might see some of that here. And you can look at that in terms of the dynamics of the system. but those, for our purpose at the moment, are not terribly important. So, if it's a minimal gene set, every gene in there should be essential. So if we knock out each gene, the cell should die. Okay? So that's one way we can test whether we actually have a minimal gene set. We can also ask the question, and this is more about robustness, what happens if the level of a particular enzyme was decreased temporarily? Would the cell recover? So this is some knockout experiments. This is with the system I just showed with the glucose 6-phosphate being converted to fructose 6-phosphate, phosphoglucose isomerase. And this we're measuring ATP, we can measure different outputs. So if we have a 25% reduction in that level of the enzyme, there's a perturbation and the cell recovers back to a stable state. If we have a 60% reduction, the cell dies, it can't recover. And again, if we do the knockout, the cell dies. We can also ask questions about how this is related to the environment. We have an environment where we had to supply all the amino acids because we didn't have ways for the cell to synthesize the amino acids, for example. We had 31 different components in the external media that had to be taken into the cell explicitly. But we want transporters that are fairly promiscuous. So for the amino acids, we have one transporter that would take an arginine, histidine, and lysine. So these substrates compete for the transport. So we start with some initial concentrations corresponding to what we thought the composition of the amino acids would be inside, the proteins would be inside the cell. But then we ask, what happens if we change the ratio of those amino acids? So we see if we increase the amount of arginine 5-fold, and we let the lysine level inside the cell, it decreased, but came to a stable state. We went 10-fold, it was still stable, although at a very low level. We went 15-fold, the system died. It's not a change in the genetics, but a change in the external environment, which could lead to a system that would not function. Those are examples of some of the things that we could do with the system. What we have shown, I think, is for the first time, this possibility of a genomically and chemically complete growth model that's capable of indefinite growth and replication. And that reconstructing life in vitro is plausible. Now, a number of things we'd like to do. The physical configuration of the cell is not fully accurate, etc. But I think it shows significant progress there. The other type of model we've been doing is modeling humans. Okay, and we're not brave enough to try to model humans with a simple mathematical model. Although we base our model on physiology-based pharmacokinetic models that have existed in this community since the 60s. Okay. What we're motivated for, in part, is if I look at the drug development process in this country, it's very, very expensive, and it's not very effective. 1 in 10 drugs makes it through the clinical trials to a product. Over half are failing now in phase 3, which makes it extremely expensive. Failing often for some fairly subtle reasons. If we had a model system which allows us to be successful 1 in 4 times rather than 1 in 10 times, that has a tremendous impact. And the system I'm going to build is not going to be perfect, but if we can move us from 1 in 10 success rate to 1 in 4, it has significant impact either in terms of the cost of drugs or giving society back 2.5 times more drugs for the same investment it's making now. Either way, we should all be better off. So part of the question we ask, can we reduce our dependency on animal and animal models? We use animal models a lot, they're expensive, they take a long time, there's ethical issues are raised, but the biggest problem with animal models is when you get done with it, it doesn't really tell you very much about what's going to happen in humans. So we want to make a realistic human surrogate which would reduce our dependency on animal studies. And hopefully when we give us a better estimate of what's going to happen when we take that drug into clinical trials. We also motivate ourselves by trying to take advances in understanding the toxicity at the molecular level and incorporating that into a cell model which would be embedded in an organ tissue model which would be embedded in a physiological-based pharmacokinetic model so we could go across scales that way. So many of you, I think, are familiar with physiology-based pharmacokinetic models, but if you're not, you take the body, divide it into different compartments. You treat each compartment as really a chemical reactor, an absorber, or a holding tank. And you can predict from that the distribution of a drug in each tissue in time, and also all the metabolites. There's also what's called pharmacodynamic models, which ask if this is the concentration near an organ, what is the response to the organ, and that can be built into the system. Now the problem is, I'm not going to get my drug approved by FDA based on my model. As much as I love my model, it's not going to happen. And the reason it's not going to happen is there's a lot of secondary and tertiary effects that occur in the body I just don't know enough about to put in the model. Okay? So we were asked, could we improve the reliability of these predictions by building a physical in vitro model where we're substituting living cells, living tissue constructs for the differential equations, where these constructs can tell us about the things we did not know enough to put into the model to begin with. So this is what we've done. This is the essence of the idea. We take a conceptual model of the body, build a physiology-based pharmacokinetic model, and then we make it in a micro-fabricated format. We originally called it a cell culture analog system. It was called Body in a Chip, a little article about 6 years ago in Newsweek. So if you're going to build something like this, what would be the important design criteria? What would you have to worry about? Well, the first four are relatively easy. You want to have a realistic ratio of cell mass to one another. You want to have a liver and a lung that look like the liver and lung in the body in terms of the relative masses. I want this blood to go to different organs in the way it goes to the body. 10% goes to a particular organ. I want to build a system where 10% of what I'm recirculating as a blood surrogate goes to that organ. I want the residence time of that fluid in the organ to be the same as in the body, because that should control the overall reaction kinetics. I'm worried about physiological shear forces, because it's not only chemical interactions, but physical mechanical interactions which are important to respond to many of these cells. You want the ratio of liquid to cells in the compartment to be physiologic. If I'm using 2D cell cultures, I can't do that very easily. But if I'm using tissue constructs, 3D tissue cultures, I can do that. And finally, this is the hard part. This is where we rely on a lot of our friends, is we like the biological constructs we put into the system to be as authentic as possible. If you were to give me part of your liver, that'd be nice of you. I could put it in the system, but it would still change in its characteristics after it's out of your body for 1 or 2 hours. So building authentic constructs is difficult. So I'm relying a lot on my friends that are doing tissue engineering to give me authentic constructs. I'm relying on a lot of friends that are doing stem cell kinds of things to be able to generate more realistic constructs that we can put into this system. Most of what I'm showing you data has been with cell lines, which we recognize as not being optimal, but it's a way for us to test whether this idea is feasible or not. What are the advantages of this system compared to other in vitro systems? One thing is right away, unlike any other in vitro system, you get realistic dose dynamics. The dose of the compound, if we have a good system, should follow what it does in the body. We can actually compare what we see with the PBPK, and by doing the comparison between the model predictions and what we see, we can come to a better underlying understanding of the mechanisms that occur in the body. And since I know the number of cells, I know the enzyme levels, I know the flow in the system, if I can't predict what's happening, it means that probably my understanding of the mechanism is incomplete. In an iterative way, I can come to a much better understanding of the underlying mechanism. And finally, the big problem has been if it kills a rat, what does it mean to a human? And this kind of system can help us in the process of extrapolating data from rats to humans. So there's some challenges in doing this. This is the first micro-fluidic device to have full recirculation of flow for an extended period of time. Okay. If you look at your body, the blood in your body turns over, let's say, about 10 times per hour. So over 96 hours, that's 960 turnovers. I'm working with less than a couple hundred microliters of fluid, which will evaporate very quickly. I have to do this in a way in which I'm not getting gas bubbles, because gas bubbles will kill the system. So there's a lot of engineering challenges in simply making a system which can function for a long period of time. And 96 hours is typically what we do. If we had an artificial kidney, we could extend that period of operation, because what limits us right now is not the mechanical issues, but the buildup of toxic waste materials. We have to do in situ data analysis. If we're going to operate this system, we'd like to be able to actually take measurements with time, integrate that into a system where we can understand what's happening as time goes on. We can sacrifice the system if we want to and get data as endpoint data, but it's nicer to do it if we can do it as we go along. So it requires sensitive analytics, and we've used largely optical methods in our group. We worked with Jay Hickman, who has systems which are constructs which are electrically active, which allows us to monitor electrical activity. But there are other more advanced systems that we want to put on the system which allow us to get a better idea of what's happening. But this is one of the challenges we have. The other challenge, if you've done any tissue engineering, you know that tissue A and tissue B kind of like different media. We have to have a common surrogate. We're recirculating the same media through the system. So we have to have something that all the cells are compatible with. Again, we're not necessarily growing the cells. We want the cells to survive and be metabolically active in the system. And also we want to emulate blood, because a lot of the compounds we work with are only sparingly soluble, and the presence of serum albumin and other things in blood make a difference in the presentation of that compound or its metabolites to the tissue. So we have to emulate that as well. Again, our biggest barrier ultimately is the authenticity of the biological constructs. So if we look for biological constructs, obviously want something that mimics the biological response of our normal tissues, that's functional, like a neuron's functional, neuromuscular junction is functional. Because of limits of fabrication, there is an advantage. It's not an absolute requirement. It's an advantage that if we work with 200 microns in depth is better. That would be probably about 20 to 30 microns per flow. It means we have a construct that's about 170, 180 microns in size. But that's enough to emulate a lot of the things that occur in the body. If you look at the body, distances between capillaries for metabolically active tissue, it's about 200 microns, because the penetration depth of oxygen is about 100 microns. So it's consistent with that. We ultimately would like to have a device that can be stored, pulled off the shelf, reconstituted quickly, slapped down, and used in drug discovery process. That's one of the other things that we've been working with. So design and assembly. Now I could build this with 1,000 different valves in it, but we chose not to. We chose to use passive flow control. So what we have to do is we know where the inlet is and the outlet is. I don't have a pointer, but there's an inlet and outlet here. And all we do is calculate what the pressure drop should be between inlet and outlet. We size the particular conduits to give us the break and flow that we want. If we want 10% of the flow to go down this way, if we know the resistance to flow and we make the cross-sectional area of the channel appropriate, we should get 10% of the flow going down this way. And what the table shows you is that what we calculate turns out to be pretty close to what we measure. For example, in this particular model, we have a tumor compartment. We predict the residence time to be 70 seconds. We measure 69.7 plus or minus a bit. We want the velocity to be 71.5 microns per second in the system, because again, we calculate shear rates and other things like that. And we measure 68.9 plus or minus a little bit. So you can see that what we predict and what we can measure is reasonably close to one another. So this method allows us to actually, in a fairly robust way, in a fairly simple device, be able to fairly accurately predict what the residence time, etc., is going to be. The chip I just showed you is not the whole body. It's the compartments where we're worried about reaction or absorption occurring. The rest of the body, in some sense, acts as a holding tank. We cheated a little bit. You have to go back to the PPK and see how we cheated. And we can make corrections because we know how we cheated. But let me just say, we lumped all the other tissues in this medium reservoir debubbler. This is probably how we control the buildup of gas. We get rid of gas from the system. We use, in this case, a peristaltic pump, but we do have a pumpless system. But most of that I'm going to show you today is from a system with a pump built into it. So let me just give you a couple of quick examples. So we want to predict human response to multiple drugs for treating cancer. As you may know, multi-drug resistance is an important problem in treating cancer. You treat a cancer, you give it a drug, the cancer more or less disappears, then it reappears again, and is resistant not only to that initial chemotherapeutic agent, but a whole range of chemotherapeutic agents. I'm going to worry about kind of multi-drug resistance occurs from overexpressed in P-glycoprotein, which is a pump protein on the surface of the cell. The cells become multi-drug resistant by making 50 to 100 times more of this pump protein, so you can't get enough drug inside the cell to kill the cell without wiping out the rest of the body. So we're going to use a very simple model. We're going to use a liver because we're always interested about not only the compound we deliver, but its metabolites. We use bone marrow representing kind of a normal tissue. The megakaryoblast cell line is important for platelet formation. And then we're going to use as our target uterine tumor. We have a parental line and a derivative cell line which is derived from it and presumably is different only in its overexpression in P-glycoprotein. It is derived as being resistant to doxorubicin. So we're going to use as a model doxorubicin as the chemotherapeutic agent, which also has the advantage that it's naturally fluorescent, which allows you to measure concentration inside and outside the cell. We use cyclosporin, nicardipine as MDR suppressors. Cyclosporin is an immune suppressor, nicardipine is a beta channel blocker. Both have been used in clinical trials as MDR suppressors. Both have failed because they cause too many side effects. Not because they don't work, but they get a concentration sufficient to work as MDR suppressor, they cause too many side effects. So this is just an example of some data. The blue bars are what happens if I don't drug this system. And I find, and this is a little different than what I do most experiments. We seeded the rather low-level cells and looked at the proliferation of cells. The liver and the bone marrow compartments grow by about 3.5. The two cancer cell lines grow by 5.5. Then we add doxorubicin to the system at a level which is not terribly unrealistic. It's about 2 to 3-fold higher than what would be used clinically and based on area under the curve. Then we add doxorubicin with nicardipine, doxorubicin with cyclosporin. And what you should see, and I would show you with a pointer, but if you look at the MDR component, you notice that there's a significant response when we add the MDR suppressors, and none of the other cell lines show a response to MDR suppressors, what we expect, with overexpression of P-glycoprotein. And what's most interesting here that we see is if we take half the normal level of cyclosporin, half the level of nicardipine, we get a synergistic interaction which further reduces the response in the system, further reduces proliferation MDR system. Now that doesn't mean that you should go out and prescribe these compounds to somebody who has MDR-resistant cancer, but I think what it shows is we're building a system which could allow you to look for such compounds, and that's what we want to do here. The synergism is not observed in well-played assays, which would be the common way of doing this, and we think, again, this might be a device to be useful to probe various combination therapies for cancer. Similarly, we've done something with another device. We've been interested in treatment of colon cancer with Tegafor. So this is a treatment that works well in about 15% of the population, not so well in the rest of the population. The reason for that, I'm not going to have a chance to go into today, is there's an enzyme dihydropyrimidine dehydrogenase that varies 20-fold in its level of activity in the population. So if you produce a lot of this enzyme, you actually get much active material. If you have a rather low level, you get a toxic effect basically from the same dose. Tegafor is a prodrug, has to be converted by P450s, primarily in liver, to 5-FU, and you put in uracil as an inhibitor of the DPD enzyme, which should allow the 5-FU concentration to remain high enough to be effective in the system. And the 5-FU interferes with DNA and RNA synthesis and thymidine synthetase, so basically nucleotide synthesis. So again, we built a system, liver metabolism, colon cancer is our target, and we had a different bone marrow myoblast cell line this time as the model. And the megakaryoblast we chose last time because it was attachment dependent. Here, this system is suspension dependent. And that's because instead of doing tantrum dependent, we're putting things in a 3D hydrogel to make the system a little bit more realistic. So we can use Matrigel for the colon cancer, liver cell lines. You put it in, it's a liquid. You have 4 degrees C. You warm it up to 37. It gels. It shrinks. It provides a flow path. Works actually pretty nicely. If I put the chemosity cells in there, they just crawl right out of the Matrigel, so you need a stiffer gel. So we use alginate for that. Gives a little different flow pattern, and I'm going to have a significant oxygen concentration gradient in that system, which may be useful in some cases, but that's a consequence of that. Again, we build the system I talked about. We insert an extra little bubble trap. Basically, if I take a 96 well plate, the traditional assay, and I took the colon cancer and I put Tegafor on it, I would not know that Tegafor was a drug because it's a prodrug. And there's actually a slight amount of P450 activity in the colon cancer cell lines, but not very much. So I really don't notice that Tegafor is an active drug. I do notice that 5-FU is an active drug. If, however, I put it in the device that we have, we see right away that Tegafor is an active drug. That's the second bar. If I remove the liver, now one thing I can do with this model I can't do with the animal model. I can do a lot of what-if experiments. The what-if experiment here is what happens if I didn't have a liver, okay? Now if I take a mouse, I take out the liver, the experiment's over. In this system, I can take out the liver and run the experiment, okay? And if I do that, I find that the response of the system is significantly less than it would be if I left the liver in there. So it's confirming that the P450s in the liver are responsible for the conversion of Tegafor into 5-FU. And the final thing shows that the response to 5-FU, which is about the same as Tegafor. We can also do some experiments now. What happens if we add uracil to the system? That's the right-hand panel. And you see that if we just take Tegafor, the viability goes down to about 58%. If we add the uracil, it goes down to about 30%, okay, which is what we would expect. We haven't broken any new ground here, but we have recapitulated what clinicians do see in these experiments and some other things I haven't shown you yet. One other thing I want to mention is that the analysis on the system is important. We worked with a group from Seoul National University. A couple of people is Sungjoo Kim and Don Kim, who's now at Yonsei University. Basically, we built a simple optical system. We could put in an incubator, we could ratchet the system along, and do some measurements about what was happening in the system to monitor either metabolic activity or cell viability. We've begun some work with Jay Hickman at the University of Central Florida on functional tissues, which are electrically active. You can imagine that adds a way that we can monitor the system that we couldn't before, and we're monitoring function as well as metabolism, which I think is an important addition. The other thing we're going to ask is, can barrier models be incorporated with the micro-CCA? Because not everything you get is by a shot, right? Okay. Things that cross not only through the GI tract, but the lung, the skin, blood-brain barriers, another barrier tissue we worry about. There's other ones in the body. These control access of drug into different compartments. So to mimic oral uptake, inhalation, absorption, etc., we need this built into the system. One that we have worked on is the GI tract model. I think we have a very good GI tract model. We worked with Ray Glahn over in the USD lab on the Cornell campus. He has a fairly early passage of Keiko-2 cells, which actually are better than most Keiko-2 cells you can get. We mix it with goblet cells or mucus-producing cells, which will produce a mucus layer. So if you look at inside your gut, not only does a particle have to, or a drug have to penetrate the cells, they first have to penetrate a mucus layer to get to the cells. What this does is emulate the tightly adhered mucus layer. The loose mucus layer that would be in your gut is not emulated in the system, but we can mimic about the 10 micron thickness of tightly attached mucus. And then we can add M cells to the system by co-culturing Keiko-2 and Raja B, which are important for the uptake of particles, and one of the things we've been interested in is the response to nanoparticles. So we build a system, we have a chip, and we have a separate component for the GI, basically shown experimentally here, or diagrammically here. We culture the cells on the membrane. We have kind of an inside and outside. What we're looking here is acetaminophen. Okay, so hopefully no one's thinking of suicide. But if you were thinking of suicide, don't do it with an overdose of Tylenol, which is acetaminophen, because it's a very painful way to go. But about 100, 150 people do that each year, plus the number of the people take overdoses of acetaminophen. It was an interesting target for us to look at in this case. So we could build a model system. We put acetaminophen in the gut. Now, when we build this gut, we can actually put through an artificial stomach if we want to. Ray's developed an artificial stomach, so we can ask what happens if you take acetaminophen from rice and beans, or you do it with beef, or some other things like that. So we model all those kinds of things. And we look at what crosses this barrier and is circulated through the systemic circulation. And we're particularly worried what happened in the liver compartment in this case. So what this shows is a dose-dependent response to exposure to acetaminophen. These are all very, very high levels, far beyond therapeutic. There is data available in a mouse model of 27 millimolar acetaminophen, which looks very similar to what we see here in terms of its response in the liver. Now, if I added this just directly to the liver, I'd see a different response. Actually, the presence of the GI tract modulates the presentation of acetaminophen to the liver. Also, there's a little bit of P450 activity there, which also modifies it slightly. So if I use just the liver component, I wouldn't really get quite the same picture as I do using the system together. So a lot of these ideas are in patents. They're owned by Cornell. They've been licensed to a little company called Urel. I'm on their scientific advisory board, which has met once in 6 years. And I was there by phone. They have a joint scientific collaborations with Schering Plough. I think the European cosmetics firm is now known as L'Oréal, which I can't pronounce. And they're really kind of in beta site developing this kind of system. So again, many thanks. First of all, thanks to all of you for listening. Thanks to the committee. I am again very humbled by their selection. A big thanks to my grad students and postdocs, because they've really been, in many ways, my most important colleagues, the ones where we share a lot of ideas back and forth. Many of them have gone on to do very great work, have very great careers, and I'm very, very proud of them. I also might mention my wife and family. Many of you also have the same situation where this can be a fairly demanding profession, so you need an understanding wife and family. And also to my colleagues, both at Cornell and elsewhere, which have been very supportive, and it's been a great experience for me. So with that, I'd like to thank you for your attention. And I don't know if we'll have any questions or not, but I'd be very happy to for this time. So thank you.
We do have a few minutes for questions for Dr. Shuler. There are microphones here in the center aisles. If you have any questions, we'd be glad to entertain them.
No question.
I know people are shy in a large group, so.
Dr. Pettis.
Except Nicholas.
Absolutely great work, Paul.
And I don't think most people know that you're also participating in the Alcophysics, the NCI initiative. You have a U54 as well. The work with Eckert cells and HC29 is really incredible. It's the first time that I see somebody really applying this co-culture in more realistic systems to really see how it works, because we and others in Sweden and so on have done it, but in a very primitive way. I hope the data will show the viability at the same time they will show the potential transport. You're working with acetaminophen. I think it would be better to work, and I know you know it, with doxorubicin and other anti-cancer agents.
Yeah. We're getting to the point where I think we're like to work with more realistic problems. We pick kind of compounds for which there's some data that we can do some real comparisons, but we're interested in really moving to more interesting systems where there's less knowns.
quick questions. One is, how are you, if you have ever planned to use this system to study communication between different organs? So if there is a way you can use and model this to understand how, I don't know, liver talks to lungs. And if you're planning to use this for metastasis and study how metastases work.
Okay, so the first question, actually, that's involved in the system because we have a common flow. Anything which is released by one compartment goes to another compartment. So some of my original work was naphthalene toxicity, which is, naphthalene by itself is not toxic, but it's converted to a toxic component in the liver, and its primary effects on the lung, which we could show in this system. When we originally started, people thought naphthalene epoxide was the reactive agent. What we could show with the system was actually naphthoquinone was the reactive agent that was generated in the liver, which would have an effect on the lung compartment. But this interaction of two compartments, if it's by soluble species exchange, is something that we do capture, and we think we can capture fairly realistic. The other thing about that system is you can show that there's actually a fairly narrow window in terms of conditions in which this occurs, and it's going to be different in mice and rats, just with some differences in pharmacokinetics. So those are things that we can build into the system. The second question was what again? Metastasis. So we have not used the system to look at metastasis. One thing I did not talk about that we're doing as part of our U54 is we're building a model of the microvasculature using techniques from micro-fabrication. And we're looking at how endothelial-lined microvessels respond to changes in the fluid dynamics, respond to changes in chemical signals, at least for the first step in metastasis, where circulating tumor cells will interact with endothelium. And part of it's just fluid dynamics. A little partial blockage downstream can make a big effect on where the cells will actually physically arrive. We haven't done the part where we add the chemical signals yet, but that's something that we intend to do.
So on behalf of the Biomedical Engineering Society, it's my privilege to recognize Mike's excellent work with a plaque and an award.